So it's my great pleasure to introduce Dalphy, and I'm going to do this correctly, Harteveld. She serves as the postdoctorate assistant at WSU Northwest Rec. You heard from her boss, who is Tobin, previously, and Daffy's going to talk a little bit about her work at WSU Northwest Rec. Please help me in welcoming her.
Thank you. So I arrived in the US about a year ago, and I started working on one of the main diseases of blueberries, in specific to the Pacific Northwest, being mummyberry. And so I'm going to present what I've learned so far about this disease. So for those who don't know, what does mummyberry look like? Mummyberry appears as these browning of the flowers with some fluffy mycelial growth on the surface, and these symptoms are referred to as flower strikes. On the leaves, the symptoms are called shoot strikes. They develop as browning of the leaf veins, and then the browning spreads over the leaf blade. And in fruit, the infected fruit appears as the shriveled, whitish, dried out fruit on the plants. Now there is another stage of infected fruits that you can't really detect, so you can't really see if the disease is occurring in the plants, but it is internal. This is at the green fruit stage, and these photos I just like to show you how that looks like. On the left, you see photos of fruits at different stages, and then on the right, I cut them open, and you can actually see the white fungal growth inside of the fruit, which sometimes you can't detect from just seeing it growing on the fruit just yet. So if you want to detect if you have mummyberry infection in your fruit at an early stage, this is a way to catch it early on. So the pathogen is called Monilinia vaccinii-corymbosi, which is a fungus. And here are some photos of how it looks like. In the field, it appears as small mushrooms developing from old berries on the ground. On the plants as this fluffy mycelium on the symptoms, and when we take it to the laboratory on the culture media, it appears as this white fluffy growth. So what is the problem with mummyberry? It's causing annual losses for the blueberry industry in the Pacific Northwest. Control options are available for both the conventional and the organic industry, but the results are inconsistent, and the timing of fungicide applications, again, is calendar-based and not based on the disease biology, which could be a reason that the control is not efficient at this stage. Only a little bit of disease is sufficient to make it leading to dramatic losses in later years. There was no information on the biology of this disease for this region, and this could also be a reason that the strategies used in different locations in the US or overseas may not be effective in this region, and there's no information on the level of fungicide resistance of this pathogen. And so for my work, I focused on to get a better understanding of the biology of this disease, to be able to develop improved management options. Looking at the disease cycle of mummyberry, it's well documented. So we start with the dormant state of old berries that are lying on the ground in between the blueberry plants. In spring, under the right conditions, little mushrooms called apothecia develop from these berries. Within these berries, a type of spores is produced called ascospores. These ascospores are transferred by wind, landing on the emerging leaf and flower tissues where primary infection takes place. This the symptoms that I just referred to, the flower and shoot strikes, develop. On top of these symptoms, a second type of spores is developed, which is called conidia. Now, these conidia have a characteristic of producing a sweetened scent or nectar that mimics that the bees likes. It mimics the nectar, and so bees land on these conidia and take them into the flowers, where the fungus infects the flowers through the stigma. Then this fungus develops inside the fruits and leading to discoloration of the fruit, and the fruit drops off the plants, landing on the ground, and the disease cycle is started. So now, looking at this disease cycle, in order to prevent any infection to occur, the most important step is the primary inoculum release. So this is the event that I chose to focus on to get a better understanding about. Now, this primary inoculum release exists of three major events. So this is the germination of these mummies. With germination, I mean like the start that these apothecia start to grow, apothecia development, and the ascospore production. Reading up about what actually happens with these mummies in their different stages, here from the left to right, you see the different stages of these mummies, from the formation, dropping on the ground, turning into the dormant mummy that you can't do anything with. It doesn't grow. There's nothing that you can do with the fungus at that stage. Then germination looks as in this third picture, where little so-called stipes appear, and from those stipes the apothecia develop. Now, in order to go from this dormant stage to germination, there are several conditions documented that are required. And these are the requirement of 1,200 hours of the so-called chilling, which is a period of temperatures between 32 and 44 Fahrenheit, plus a minimum of about 7 days of over 44 Fahrenheit. So these are conditions needed for these dormant mummies to be able to develop these mushrooms, which will be important later in this talk. Now, looking at literature of when these apothecia are found in the fields, I found that in the different locations in the US, there's a wide variety of timing, starting from beginning of March to end of March in different locations. And there was no good information on what the factors are that are involved in the timing of this apothecia development. In this graph, you see the timing of apothecia found in a field in Corvallis in Oregon. This data was obtained from Jape Scheid of Oregon State University. And you can see that the range of this timing that the apothecia were found, it ranges from mid-March to early April. So with that, I determined to focus this project on determining the timing of this primary inoculum release in the Pacific Northwest to identify significant environmental factors that may affect this apothecia development, and then also looking at the cultivars in this region and when cultivars expose their susceptible tissue for these ascospores. And with this project, we aim to develop a model that can help in predicting the primary inoculum release for specific locations and to provide management strategies for different cultivars. So for the rest of this talk, I will focus on the three main parts involved in this primary infections. So this is the famous disease triangle. If any of these factors are missing, the disease won't occur. So it's important to know, to get a better understanding of each of these three factors to be able to to develop better management options. So I'm going to start with the timing of the primary inoculum. So in order to determine the timing of primary inoculum release, 2 fields were selected with a history of severe disease, one in Whatcom County and one in Skagit County. And I placed 3 so-called mummy nurseries in each field site, which was pretty much just a bucket where the bottom of the bucket was removed, and about 50 mummies were placed in these buckets that were used to monitor for apothecia development. Then to determine when ascospores were released in the field, I used spore traps and petri dishes to capture these ascospores. And in these photos here on the right, you see what a spore trap looks like. So a spore trap uses an airflow that captures the ascospores onto a rotating tape that is covered in petroleum jelly. Then I take these tapes to the lab and determine the ascospores on the surface. And that's it. The ascospores look like this photo in the middle once you see them under the microscope. So the results of this experiment showed that apothecia, the mushrooms, were found from beginning of March to 22nd of March in Whatcom County, and from March 10 till March 20th in Skagit County. However, the spore traps showed that ascospores were found from beginning of March till beginning of April, showing that my mummy nurseries did not represent what was actually happening in the field. Because if I can capture ascospores in the field but not see the apothecia, it shows that my mummy nurseries are not representative. So I will need to change my strategy for this coming season to get a better better indication of when the apothecia are occurring in the fields. Important from this is to keep in mind that ascospores were found in a long period from beginning of March to beginning of April. Moving on to the second part of the study is to determine the most significant environmental factors involved in apothecia development. In order to determine these, I used data loggers placed in the field and also the ag weather net. And factors that I included include minimal, average, and maximum temperature, average relative humidity, average leaf wetness, average soil temperature, total precipitation, and solar radiation. Now, I also tested other weather factors, but these ones remained to be the more significant ones to have a further look into. So performing several analyses, including a regression analysis for the best subset of climatic factors that are most significantly affecting this apothecia. In this table, you can see the results of the analysis, where if I included only one factor in the model, average soil temperature was the most significant factor, influencing or affecting about 31% of the variance. Whereas if I included more factors in the model, average soil temperature, total precipitation, and solar radiation explained about 43% of the variation observed. So these factors I kept in mind for a further analysis of how these environmental factors actually affect apothecia development, because now that we have the indication of which ones, we still don't know how. So to have a further look at that, I'd like to show you following the graphs. So in these graphs, you see the apothecia data indicated in yellow. When they're up the top of the graph, it's when the apothecia were present in the field. In blue, you see the average temperature, and in red is the solar radiation levels. So if you remember in the beginning of the talk, I told you how these mummies need 1,200 chilling hours and about a week of above 44 Fahrenheit. I didn't include the 1,200 chilling hours in here, but I calculated when this amount was reached. And then In blue, in the average temperature curve, here you can see those 7 days of above 44 Fahrenheit. And these were observed roughly a week before the apothecia were observed, indicating that there's still other factors required for these apothecia to actually show up. Well, since solar radiation was indicated as one of the significant factors, I looked a little bit closer into this, and you can see that from the time that the apothecia were there, you can also see that the solar radiation started reaching maximum peaks of about 14 megajoule per square meter, indicating that this may be a requirement for this apothecia to appear. Moving on to the next factors, soil temperature and total precipitation. So again, the soil temperature indicated here is indicated in blue, and total precipitation is indicated in the red peaks. And looking at the soil temperature, again, in the period where the apothecia were found, soil temperature started reaching temperatures of above 50 Fahrenheit, which also could mean that temperatures of above 50 Fahrenheit are required for apothecia to develop. When it comes to precipitation, at this point, I can't say anything else than that precipitation is required or needs to be there for apothecia to develop. Now, so this is the conclusion as we have it now of what we found for the climatic factors that may be involved. So these factors I also used to analyze the data obtained from Corvallis. There's 10 years of data that we obtained from our collaborators, Jape, and looking at the weather data of that location and their apothecia appearance data, I also found these factors to come up on the similar way as I just explained to you. So with all that information together, we will now develop a preliminary model that we will use to see if we can predict when the apothecia are gonna develop this next season, as in Wacom, as Skagit, as in Corvallis, and probably in BC, to just see how well it's gonna work for us to predict in the different locations within the Pacific Northwest. Now, moving on to the third and last factor involved in the primary infections, which is the susceptible host. In highbush blueberry, we know that there's specific stages of the crop needed to be susceptible for mummyberry. And for the flower stages, I indicated that in the tables on the site. You see the different stages in flower bud development. And in red, I indicated the stages that are known to be the susceptible stage for mummyberry. And for same story for the leaves. So the leaves are susceptible when about 6 to 13 millimeter of green tissue is available. Now, so what I did is I wanted to know when the susceptible tissue is available in different cultivars grown in this region. So I chose Blue Crop, Duke, Liberty, and Draper, and chose 3 fields of each cultivar in both Whatcom and Skagit County, marked 6 shoots on 6 plants, and did weekly monitoring of the development by taking photographs and then determining the stages on the shoots, or like the stages of the leaves and flowers. Now, results of this experiment are shown in the graphs. So the timing in these graphs that I show here is the first graph is the flower buds, the second one is the leaf buds. The time frame presented is the timing that the ascospores were present, and what I like to amount of susceptible tissue is demonstrated. So what I like to show here in the different colors are the different cultivars. In the stripes, we have Whatcom County, and in the continuous lines is Skagit County. And we found that overall Liberty was having the highest amount of susceptible flower buds. And looking at the peak, the timing of peak amounts of these susceptible flowers, we found that Duke and Blue Crop have their peak amounts of susceptible tissue available earlier in the season compared to Liberty and Draper. Well, this may be important if we know that the ascospore release is taking place earlier in the season or later in the season as well, as that may mean that these cultivars are more or less susceptible at that stage. Looking at the leaf buds, there was a lot of variation observed, but between the 4 cultivars, it showed that Blue Crop had most of its susceptible leaf buds available earlier in the season compared to the other cultivars. And this graph shows the total amount of susceptible tissue found on the plants over the ascospore period. And well, the biggest difference that I like to show here is that there was way more susceptible flower tissue found in the plants compared to leaf tissue, indicating that flowers may be play a more significant role in this disease, as well as in management compared to leaves. And you can see between the 4 cultivars, Liberty had the highest amount of susceptible flower tissue, then Draper, Blue Crop, and Duke, whereas for leaves, Blue Crop had the highest amount of susceptible tissue. So for this first year, the preliminary conclusions I'd like to share is that my mummy nurseries were not representative for the apothecia development in the field. We found environmental factors that play a significant role in the apothecia development, including soil temperature, solar radiation, and precipitation. We found that the timing of flower shoot tissue emergence may play a more important role than timing of leaf tissues, also indicating that protecting your flower tissue may be more important than protecting the leaves. And that Duke and Blue Crop have more susceptible tissue earlier in the season compared to Liberty and Draper. For this coming season and the second year of the study, we now are working on this model to help predict the apothecia development for this coming year. So we will do the same data collection that I just presented today to validate and optimize this model. Then by the end of 2016, we will have this preliminary prediction model ready to be adopted in growers management plans for 2017 and more confirmed management recommendations for different cultivars. So with that, I'd like to thank the Washington Blueberry Commission and the Washington Commission on Pesticide Registration for funding this project, as well as the Washington State University, and our collaborators at Oregon State University, Len Coop, Jabe Scheid, and Jade Florence. So thank you very much.
Do we have any questions?
Yes. One is what causes the spores to volatilate or to go up? What causes them to lift?
So yeah, the question is, how does the spores get in the plants?
From the ground up.
Yeah. Well, what we believe now, it's actual wind.
What?
Wind.
Wind.
Yeah. Yeah.
Another question is if we had a hard freeze and up here in the Pacific Northwest, would that kill any of the so-called mummyberry spores that's on the ground if we had a hard freeze?
If you had a hard freeze, well, like early spring, I think it will slow down the further development of the apothecia, but it doesn't kill it. No. Yeah.
Dalphy, how are you going to change your mummy nursery? What do you got in mind to make some changes?
Okay, so the question is, what do I have in mind to change my mummy nursery plan? I think that I will I will use about 100 or 200 mummies and spread around the field, individual mummies, and look at those rather than just coming back to 3 spots in the field, going to like a whole wide range of like covering the whole field. Yeah. And if I look at individual mummies, I can also see how long each mushroom or each mummy is active for that sense, in that sense. Yeah.
It was mentioned that you need 7 days of temperature above 44 degrees Fahrenheit. Is that consecutive days or is that cumulative days?
I'm not sure actually, but from what I found and what I read, yes, it should be consecutive days.
Thank you.
Yeah.
Any other questions? Going once? Going twice? No more questions. All right. Thank you. Please give Dalphy a big round of applause for her presentation. Excellent. And then this is the end of our session. If you would please remember to get your pesticide credits over to your left, and then also turn in your surveys so that we can get those results back and improve upon our workshop for next year. Thank you so much.
As a reminder, please turn in your surveys for this session. There's a box at the far end of the room here for your surveys. Thank you.